All right, I am recording and I will post this to our Canvas site later today. So, like I said, this is going to be nice and short and sweet. I have another student dropping in. The first thing that I'm going to talk about is the structure of a nerve itself. So it's just a physical conversation. All right, so getting all that audio business taken care of. So when we talk about what is a nerve, let me call it my whiteboard. This is the word that we use all the time. I damaged a nerve, I pinched a nerve. So let's just look at the actual structure of a nerve itself. We call nerves typically, we use this language, cranial or spinal nerves. Those are just names given to nerves based on where their derivation is, where they originate. So do the nerves originate in the cranium, the brain, or the spinal cord? So when you hear or read cranial nerve, spinal nerve, that reference is just about where that nerve originates location-wise in the human body. Technically, the definition of a nerve is just I'm just writing a bundle of fibers, in parentheses, that means axons. It may or may not be myelinated. So typically a nerve itself, that's supposed to be S-O-M-E. I'm just writing quickly. Some myelinated, some not. Enclosed in connective tissue. So a nerve itself, what we call a nerve, is just basically a bunch of fibers, axons, bundled together. Now they're not typically all the same, but let's look at the anatomy of this and the connective tissue coverings around them. And the good news is all of this language is going to sound very familiar because they're basically arranged like a muscle is arranged. So we have endoneurium. Remember we used terms endomysium, perimysium, and epimysium. Endoneurium is just a thin, delicate connective tissue that wraps the axon of individual neurons. So endoneurium is just analogous to endomysium in a muscle. And we have perineurum, which is a thicker connective tissue that wraps several axons or fibers of neurons that are already individually wrapped in endoneurium to form, guess what, same word, a fascicle, which is a bundle of individual neuron fibers or axons. And finally, there's epineurum, which wraps, it's very coarse, sorry, let me write that first. It's coarse connective tissue that wraps several fascicles to form the nerve. So there you have it. That's what the anatomical breakdown is of a nerve itself. There's other tissues besides just what I've listed within a nerve. There are blood vessels, a lot of connective tissue, lymph vessels, and so let me just write that, includes vessels, connective tissue, blood vessels is what I meant first by vessels, so I'm going to specify lymph vessels as well to make this structure of what we call a nerve. And it is actually the wrapping, the lipid wrapping around axons that we know as myelin that makes up the bulk of the volumetric bulk of a nerve. So I'm just writing that myelin sheath makes up most of, we don't need two ofs, most of the bulk of a nerve. Now, the functional classification of nerves, I'm talking necessarily about neurons here, I'm talking about a full nerve based on pathway transmission. A motor nerve carries an impulse away from the central nervous system. So a motor nerve has individual neurons with axons that carry impulses away from the central nervous system. Sensory nerves, this is all familiar language to you now, so it shouldn't be difficult to wrap your head around these terms. Sensory nerve carry impulses toward the central nervous system. And a mixed nerve contains both functional types of neurons. So I think of it, when I think of these nerves, I think this is a one-way street in the direction away from the central nervous system. This is a one-way street in the direction toward the central nervous system. And these are two-way streets to and from the central nervous system. Mixed nerves, by the way, are by far the most common in the human body. Exclusively motor nerves and sensory nerves are really quite rare. Now, the fibers within them have names as well. So I'm going to give that a capital. This is just language. We're just delineating language. The fibers within a nerve, we give names based on the fibers based on their individual functions, aka effector organs, and their directional pathways. So fibers within a mixed nerve, for example, let me specify that, would be classified as follows. I'll break this down for you after I write these names. Somatic afferent fibers, let's just, somatic, just think voluntary skeletal muscles. Afferent is a directional term that means going toward the central nervous system. So a somatic afferent fiber would be carrying, it's a sensory fiber carrying information from a skeletal muscle toward the central nervous system. Within a mixed nerve, we also have somatic efferent fiber. This means skeletal muscle. This means moving away from directional term, away from the central nervous system. So this would be, I'm just going to write, this would be sensory. This would be motor, carrying a command toward the skeletal muscle, not away from it. And then there is, sorry about going to a new whiteboard, I'm still talking about fibers within a mixed nerve. We would also have fibers called visceral, afferent. Let's just break this down. This means not skeletal muscles. This usually means involuntary organs, vessels, or glands. This is a directional term meaning going toward the central nervous system. So these fibers would be sensory, carrying information from, say, our visceral organs, things in our abdominal organ or our blood vessels, carrying impulses toward the central nervous system. And finally, there are visceral efferent fibers, carrying impulses from the central nervous system toward a visceral organ, cardiac muscle, smooth muscle, or glands. So carrying a command. So these would be motor fibers. So all these different fiber types these two somatic, as well as these two visceral, can be found within the same nerve. Okay, so that takes care of the basics of how a nerve is, let's go to a new topic here. Takes care of how a nerve is structurally organized. That is not rocket science. I know you can wrap your head around that very quickly. Next topic of the day, which I think should hopefully be our last topic of the day. Like I said, I'm going to try to make it quick today. I'm going to call up a whole new whiteboard just because I want to remind you of this. This will help us. I was going to draw the map of the entire nervous system, but let's do this. Let's do injury of a nerve and what happens. So I'm going to write injury and repair of nerves and neurons. This is a fairly easy, quick topic for us to cover. So when we talk about injury of a nerve, I'm going to highlight this. Both nerves and neurons in the central nervous system are not capable of repair and regeneration. I have to tell you that there is a tiny bit of evidence right now in the scientific research literature that states possibly there is evidence of minor repair and regeneration in the olfactory area of the central nervous system, but that's way the exception in general. So it is sort of healthy and useful to remember that neurons in the central nervous system are not capable of regeneration. The tissue just isn't. I'll explain why in a minute. But if we talk about the peripheral nervous system, I'm just going to write this. Axons of peripheral nerves may regenerate. And the reason we say the word may is it depends on the degree of injury and the location of injury. So they may regenerate, particularly, especially if the damage is distal to the cell body. So if a nerve, even a peripheral nerve, gets damaged at the cell body, chances of repair are low. However, remember in our motor nerves, our typical motor nerves, if the injury occurs somewhere along this axon, which is the most common place nerves get damaged, by the way, and the injury is not super severe, in the peripheral nervous system, there is a fairly good chance of regeneration, especially if there's not a huge gap between this area. If this nerve just gets crushed or damaged somehow, the chances of repair are really quite good. Now, the difference between central nervous system neurons and peripheral nerves and neurons is mostly due to the myelin sheath and the cells that make up those particular myelin sheaths. So I'm going to put a double star here. So we know that axons of the central nervous system cannot repair. Axons of the peripheral nervous system may, and usually do, especially if the damage is distal to the cell body. The difference lies in this. Schwann cells versus oligodendrocytes. We know these are the neuroglial cells that make up the myelin sheath and peripheral nerves. Oligodendrocytes are the neuroglial cells that make up the myelin sheath in the central nervous system. And the difference between their nerve regeneration capabilities lies in the differences in these cells. Schwann cells are one of, if not the most major player in assisting nerve regeneration. I'll try to explain a few of these differences. Oligodendrocytes actually actively suppress central nervous system regeneration. So this is the key to why our central nervous system cells don't regenerate and our peripheral nervous cells do. I'm just layering over the top of this. I won't erase it. Oligodendrocytes in the CNS are covered with growth inhibiting proteins. And astrocytes, which are also found in the central nervous system, quickly form scar tissue, so that doesn't help. So these two factors, especially this one, really prevent injured cells in the central nervous system from regenerating. But let's talk about axon regeneration in the peripheral nervous system. first, I do want to tell you that there is a step-by-step. I'm going to highlight the major points of this, but I'm not going to go through read for you word for word. There's a step-by-step process in your textbook. I think it's figure 13.5. Just look for the figure that lays out the step of nerve regeneration. Here's what goes on in a more Reader's Digest version. I'm going to highlight the important points. After injury, the portion of the axon that's distal to the cell body, or the injury itself, degenerates. So we get a degeneration of tissue. That process has a name. It's named after the guy who discovered it. Wallerian, W-A-L-L-E-R-I-A-N. We call this Wallerian degeneration. So it's axon tissue that actually goes through Wallerian degeneration and dies off. Schwann cells are like magic cells. Schwann cells do not degenerate. That's key to this whole regeneration process. They actually, after injury, they start to proliferate. That means they multiply. And they form this bridge over the injured area. I'm just going to write bridge across injury. So if the axon, let's say, is damaged from injury, the distal part of if this is the cell body end, the distal part of the axons, this tissue starts to degenerate. Schwann cells start to proliferate and actually form a bridge. Not unlike, at least physically, it looks like the bone callus that's formed after bone is injured. But these are actually Schwann cells, those lipid. They form a bridge across that area. Then the cells start, Schwann cells and macrophages really work together. Schwann cells call in macrophages. So here's a macrophage. Remember, those are non-specific immune cells. Schwann cells release chemicals that sort of signal macrophages to come in and start cleaning up the dead debris. So let me write that. I'm just going to write Schwann cells signal macrophages. Those macrophages are attracted to the area and start cleaning up dead debris. And then the macrophages also secrete chemicals that stimulate Schwann cells and new axonal growth. So let's just say this. Schwann and macrophages secrete NGFs. That stands for nerve growth factor. So chemicals are secreted by the cells in the injury area, the active cells that secrete what we call nerve growth factor. And that stimulates fibroblasts in the area become active and also start to secrete nerve growth factor as well. So we get all of these cells in the injured area, the major players, Schwann, macrophages, and finally fibroblasts, secreting chemicals that stimulate new axonal growth. And then a new axon starts growing in that bridged area. That bridge that the Schwann cells made. So Schwann cells then wrap around the new axonal growth. So we have, what we had was an injured axon originally. There's a macrophage. I'm using different colors. Here's the original myelin sheath. We have Wallerian degeneration of this distal tissue. Schwann cells in the area start to create a bridge at this injured area. They release chemicals that signal macrophages to come in, clean up this dead tissue. Macrophages also secrete chemicals that stimulate Schwann cells, fibroblasts, to start secreting new nerve growth factor, which causes so let's just erase this old axon that's died off and now new axon tissue starts to grow in this bridged area to form a connection. Now here's the caveat. This regeneration almost never regrows or regenerates perfectly as the old one did. We might get nerve growth going off this way, or new nerve growth, axon growth going off this way. It's never perfect, or rarely is it perfect. So oftentimes, retraining of the innervation pathway of this damaged nerve is necessary to actually start getting function back. So what I've described here with nerve regeneration lays down the map, the framework for the possibility of regaining nerve control to that specific muscle or that effector organ. But oftentimes, especially in the somatic nervous system, the motor somatic nervous system to skeletal muscle, we need to retrain that pathway if there was significant damage done to it. So I'm just looking at my notes that I jotted down before this to make sure that I went over everything that I needed to say about, I think I did. All right, guys, so I'm gonna call it good there. That was a nice short and sweet lecture. Let me cut off my recording and then